So welcome everybody one more time to this third edition of Corpus Curiosum. This is the second talk of this series. And I think, I think most of you already know us, but let me explain a bit the project for those that are new here. So first of all, the name. Probably you already have thought about that, but we got the inspiration from this corpus callosum, that is this nervous tract that is connecting both hemispheres of the brain. And in the same way, we are aimed at connecting early career neuroscientists all over the world. And this is us. Behind the scenes, we also have 3 early career neuroscientists placed in different cities in Europe and also in different stages of our careers. We have Ines, that is a PhD student in Madrid. We have Faisal, a graduate student now living in Germany, and myself that now I'm living in Zurich. But we are not alone at all in this edition. We created the ambassador program with the goal of reaching as many countries as possible. So here you have all of our awesome ambassadors. They are coming from 17 different countries. We also have our sponsors. First of all, we wanted to share with you guys that for this third edition, we are very proud and very happy because we were able to win the diversity grant of the IBRO, the International Brain Research Organization, because one of the main goals of this project is also to show actually and to promote the diversity in neuroscience. So that's why we make sure that we have as many women as men as speakers and also in the audience and as well in our ambassador program. And we also make sure that we count with people coming from all over the world. We also count with the support of the fans from the very beginning, from the day 0 that we started this project. So we are very thankful for that. And we also count in this edition with the support of the SCFAs. They are an organization of Spanish researchers here in Switzerland. And they are all the time organizing super cool activities and also giving support to everybody that needs it. So here you have the contact in case you want to contact them. And also here you have a QR code where you can find more information about them. Finally, thank you to the hard work of the ambassadors and the promotion of our sponsors. You guys, our audience comes from 47 different countries, which is amazing for us. And also from more than 100 different companies. So thank you to everybody. Yes, also some important notes that you have to know is that this introduction and also the main talk will be recorded, but you don't have to worry at all because the discussion won't be recorded, so you guys are not going to be recorded at any time. So that's why please feel free to make questions whenever you want. You can use the chat of Zoom to write them down. And then at the end of the talk, we will have the discussion. So you have to know that this is a very safe space. There are no silly questions. So please take the time and the opportunity to discuss a bit with us and with Yoko, with the speaker, and we will have a great time for sure. And finally, regarding the length of this session, it is 1.5 hours. So that means that we are going to close the session at 4:30 p.m. UK time, but of course you are free to leave whenever you want. Now, last but not least, for sure, we have our speaker. I will introduce her. She is Yoko Wang. She's a PhD candidate in the Adelaide Medical School, the University of Adelaide, and the Lifelong Health Team, South Australian Health and Medical Research Institute. She also holds a bachelor's in biotechnology from the University of Surabaya, and she is an Australia Awards scholar from Indonesia. Her current research focuses on understanding why people living with obesity feel a stronger drive to eat and how to help them improving their eating behavior. And in her PhD, she studies the bowel-gut-brain axis of appetite regulation in obesity with particular interests on the effect of inflammation and dietary factors. And Yoko Wang will be talking today about unboxing the microbiota-gut-brain axis.
All right, first of all, I would like to thank the organizers for giving me the opportunity today to share about a broad overview of the gut microbiota and the brain axis in this presentation. And to begin with, I would like to start by a quote that perhaps many of us are familiar with, that is, all diseases start from the gut. If we realize that the research and science around the gut-brain axis today revolve around this idea, and our current understanding about the gut-brain axis, it is a bidirectional communication pathway that involves a lot of different components, such as the vagus and spinal nerve from the peripheral nervous system. And in the gastrointestinal tract, we have the enteric nervous system, enteroendocrine cells, and recently identified neuropods. So neuropods is a type of enteroendocrine cell that forms a direct synaptic connection to the vagus nerve or the enteric nervous system. Furthermore, we also have the signaling molecules, such as the gut hormones and neurotransmitters, that help to convey the messages from the gastrointestinal tract to the brain and vice versa. This gut-brain axis is really important as it involves in the regulation of various physiological regulation in the body, such as to help maintaining gut homeostasis, brain function, as well as mental health and well-being. Studies have also shown that disruption in this gut-brain axis can lead to many types of diseases. And in the past couple of years, we've got a newcomer in this gut-brain axis, that is the gut microbiota. Although they are tiny, although they are small, they have a really big role in modulating the gut-brain axis. So speaking about the gut microbiota or the gut microbiome, it's not just the bacteria. There are also archaea, viruses, parasites, or even fungi. However, research today are still focusing on bacteria. And these gut microbiota are distributed throughout the gastrointestinal tract, as you can see in the picture that the highest abundance can be found in the colon. And research about the microbiota and gut-brain axis actually were very well established in 2012, where studies using germ-free mice and antibiotic-treated mice, they found changes in behavior, changes in neurotransmitter expressions, and also in that study, they found that microbiome metabolites can actually directly or indirectly signal to the nervous system. And this initial findings built a solid ground for us to build the research around this gut microbiota and brain axis until today. And for the next question, then we will probably be wondering, where do we get the microbiota? So we get the microbiota from our mother. When we were born, our body will make a contact with the delivery canal, which this is the first time when we were exposed to the microbiome of the microbiota of our mother. And after birth, the gut microbiota in our body will continue to change in terms of numbers and composition throughout the lifespan. And studies have associated these changes in gut microbiota with different stages of brain development, as you can see in the picture. What I would like to emphasize from this picture is there are three stages or three sensitive periods throughout our lifespan. The first one is the early stages during infancy or childhood, where the initial composition of our gut microbiota can shape the long-term composition of our gut microbiome when we are adult. And we will undergo a transition period during adolescence, and in adulthood and aging period, the composition of the gut microbiota, the gut microbiome itself is relatively stable, although they are more susceptible to changes because of external factors, such as diet or exercises, for instance. And yeah, and the next question after we know where did we get the gut microbiota is, how do they actually talk to us? Like, how do the microbiota talk to the brain? And still using the concept of the gut-brain axis, where it is a two-way relationship, is a bidirectional relationship, and so does the microbiota and the brain axis. This is a very nice picture that summarizes the communication between the gut microbiome and the brain. It may look a lot of things happening in this picture actually, but we'll go through this picture one by one. So for the first one, I would like you to focus on this picture where it emphasizes the bidirectional communication between the gut microbiome and the brain through the afferent and efferent pathway. Then the gut microbiome and the brain, they use three major pathways to communicate with each other. The first one is immune system, and then we have endocrine system, and of course the neuronal pathway. And within these pathways, the gut microbiome and the brain, they use different types of signaling molecules. It can be hormones, cytokines, metabolites secreted by the microbiota, and many other signaling substance. Then the last one, this is a broad overview of what actually happens in each pathway. You can see in the immune system, the gut microbiome and brain axis, they helps to regulate microglia maturation and function. Then also, of course, the immune response in the endocrine system. It involves in stress response through the HPA axis. And also you can see there's quite a lot of gut hormones here, and also neurotransmitters such as serotonin. And the most interesting thing that I found about this within these three pathways are the neuronal pathway, because we've got the vagal and spinal root, and of course the enteric nervous system. But as you can see in the picture, this nervous system, they express quite a lot of different receptors, which put them into a position where they can sense different type of signaling molecules in this axis. And the other important thing is the crosstalk between these three pathways. And the neuronal pathway, it's kind of like facilitating all the other pathways. Like we know that vagus nerve, they also function in the inflammation response through the vagal reflex, and enteric nervous system also sends different type of gut hormones and neurotransmitters. So that's why in this talk, I will focus on the neuronal pathway in the gut microbiota and brain axis, and one microbiome metabolite, which is the short chain fatty acids. It is one of the most well-studied microbiome metabolites in the gut microbiome and brain axis. So the first one I would like to discuss about the vagus nerve, or the gut wanderer. So the vagus came from a Latin word, means wandering, and it is the longest cranial nerve in our body that goes down from the basal of the skull, goes down to the thoracic and abdomen area, where it mainly innervates the upper GI tract. So it sends the mechanical and chemical stimuli that happens in the gastrointestinal tract, transfer the signal to the cell bodies in nodus ganglia. Then the signal will be transmitted to the upper region in the brainstem and the brain. And we know that the main function of the vagus nerve in the gastrointestinal tract is to maintain the homeostasis, and also involve in inflammatory reflex through the vagal reflex. And recent studies also shown that the vagus nerve heavily involved in the regulation of feeding and reward mechanism, as well as stress and anxiety response. And current evidence in the gut microbiota and brain axis through the vagus nerve pathway, they show that that giving a single bacteria called the Campylobacter jejuni into a mouse, it triggers anxiety-like behavior. And other studies, they did vagotomy, basically got the vagus nerve connection between the gut and the brain. This vagotomy abolished the effect of prebiotics towards the learning activity in mice. And what I would like to emphasize is the vagus nerve express quite a lot of receptors that can detect different neurotransmitters and hormones, which put this peripheral nervous system into a strategic position where they can deliver the signal from the gut microbiome in the gastrointestinal tract to the brain, although current studies are still required to establish the neural circuit. And the next one is the enteric nervous system, or probably many of us know the enteric nervous system as the little brain. It's a compilation of neurons in the gastrointestinal tract wall, primarily in the mesenteric plexus and also submucosal plexus. They form a mesh-like structure where they operate independently outside the central nervous system. That's why we call this structure as the little brain. And the primary structure, the primary function of ENS is to control motility and fluid movement in the gastrointestinal tract. There are quite a lot of studies where they show the relationship between the gut microbiota and ENS. So early studies in this area suggest that gut microbiota can influence ENS development and architecture, which suggests that perhaps gut microbiota may have an impact on certain diseases where ENS is malfunction or not fully developed. But later on, other studies also suggest that ENS can actually regulate the gut microbiota composition. So currently is in a stage of like chicken and egg, like cause and effect, which one comes first, like gut microbiota influence ENS development or on the other direction. But a very interesting paper came up in 2018 where they discovered that the effect of gut microbiota in ENS development, it was through the serotonin pathway. And the third one I would like to talk in this neuronal pathway within the gut microbiome and brain axis is the spinal nerve. The spinal nerve is quite similar to the vagus nerve in terms of their ability to detect mechanical and chemical stimuli that happens in the gut. For instance, in the colon, as you can see in the picture, there are quite a lot of different type of endings, and that's in different layer of the gastrointestinal tract. They also express quite a lot of different channels and receptors. And currently, what we know about the spinal nerve is they mainly involve in pain sensation and regulation, but in the other studies, they also revealed that it actually involved in normal physiological condition. So the relationship between the gut microbiota and the brain through the spinal nerve is mainly studied in the irritable bowel syndrome. I wouldn't go too deep in this slide because I will talk more about the IBS further down the presentation. And now we go to the signaling molecule. The short chain fatty acids are the most well-studied signaling molecules in the gut microbiome and brain axis. It is a product of fermentation of carbohydrates from the intestine by the gut microbiota. And there are 3 types of short chain fatty acids, acetate, propionate, and butyrate, which can be detected through receptors, free fatty acid receptors as main recipients, but there are also other GPCRs that can detect the SCFAs. So the main producers of these short-chain fatty acids in the gut, for instance, are the Bacteroidetes, which produce acetate and propionate, and we've got the Firmicutes that produce the butyrate one. And as you can see in the picture, one contribution where SCFAs play a role in the gut microbiome and brain axis is through modification of gene expression or epigenetic modification through histone deacetylase. And this one is a very nice summary about how the SCFA, they play a very important role in different pathways of the gut microbiome and brain axis. As you can see, it involves in the immune pathway, endocrine pathway, the vagus nerve, and other humoral pathway. And it involves in the regulation of many physiological conditions and also disease pathophysiology. What I would like to emphasize in this slide is that the production of SCFAs is heavily affected by the dietary fiber, probiotics and prebiotics that influence the intestinal microbiota and subsequently the production of SCFAs. Then after we know where do we get the gut microbiota and how they communicate with the brain, then if we imagine when the gut-brain, this relationship, the gut microbiome brain axis, it goes out of balance, it can lead into different stages of health condition and changes in composition or function of the gut microbiome. We know that as dysbiosis. So as you can see in the picture, in the homeostatic condition, we've got a nice composition or between different types of bacteria. But when this homeostasis is altered, when dysbiosis happen, the pathobion can expand, then it can reduce the diversity, and then the good microbiome, the healthy microbiome, they can like reduce in numbers. Then it leads to changes in our health and well-being. And since I will mention quite a lot of diversity, gut microbiota diversity in this presentation, that I think it will be nice to have quite a bit of explanation at first. So when we talk about composition in gut microbiome, we would like to know the diversity. There are two main terminology about diversity in gut microbiome, in gut microbiota. The first one is the alpha diversity, where we look at composition within one sample, for instance, from one individual or from one mouse. And we've got the beta diversity. This is the stage where we compare the composition between two or more individuals or animal models. Then just a quick look on what diversity is. So diversity, there are two components of diversity. The first one is richness, which reflects how many types of species are there in a community. Then we've got evenness, which reflect how even is the distribution. On the other words, is how many individuals of each species in the population. And the combination between richness and evenness, we call this as diversity. Then, as I said before, when the balance of the gut microbiome and brain axis is disrupted, where this connection is out of balance, it can lead to changes in our behavior and health condition. And there are many factors that affect this gut microbiome and brain axis, such as diet, genetics, mode of delivery, environment, drugs, and exercises. And this is just an example of many types of diseases or health condition that can result when the gut microbiome and brain axis is disrupted. In this presentation, I will focus on diet as a factor, and irritable bowel syndrome as an example of diseases, because currently in irritable bowel syndrome is the only one where treatment targeting the gut microbiome using diet have shown very promising results in a clinical settings. So we talk about diet. It's like you are what you eat, and now you are what your gut microbiota eats. So it's important to understand what kind of diet that you eat and how it impacts the gut microbiome, and subsequently influence your health condition. And what I would like to emphasize from the dietary intake or diet is there are different type of source of our foods. Then within each food, we've got different nutrient components, and every day we just don't eat one type of food or one type of nutrients, but we eat like combination of foods, combination of nutrients, and it form a dietary patterns when we can see interaction between foods. And this is a very nice systematic review that I found, which demonstrate how different type of nutrient source or food source, nutrient components, and dietary patterns actually affect different types of bacteria in our body. So the first one is an example of different source of macromolecules, for instance, like plant-based protein and animal-based protein. As you can see, it influences the abundance of different types of gut microbiome and also different types of physiological changes and health condition. The similar trend can be observed also in different nutrient types. For instance, the unsaturated fat and saturated fat, where it influences different types of gut microbiomes and affect different types of subsequent physiological changes in our body. And this one is dietary patterns. What I would like to emphasize here is different dietary patterns or different combination of foods can actually have different effects on the same bacteria, which then produce a different effect also on the health status of a human, which emphasize the importance of how diet can modulate the gut microbiome and or the gut microbiota, and thus produce health outcomes. Then this one is the irritable bowel syndrome, as I mentioned before, that the irritable bowel syndrome is currently affecting 5 to 10% of population worldwide, and there are different possible pathophysiological mechanism how this irritable bowel syndrome could happen or emerge. And dysbiosis is one of the possible mechanism. As you can see in the picture, then where in this IBS condition, there is an increased sensory input, which mainly facilitated by the spinal nerve that sends signals to the brain. It changes how the CNS works, disrupt the gut-brain axis, thus producing symptoms of the IBS, altered gastrointestinal sensation. Normally they will feel pain when people have IBS, and also alter the motility and secretion in the GI tract. Then another systematic review, they showed that in IBS patients versus healthy controls, there are changes in gut microbiota. As you can see in the picture, the bifidobacterium and faecalibacterium are reduced in the IBS patient, and on the other side, the lactobacilli, quite difficult to pronounce, and Bacteroides and Enterobacteriaceae increase in IBS patient. And what's interesting in this IBS and its relationship with the gut microbiome and gut-brain axis is that the current evidence using probiotics to improve the symptoms of IBS patient, they found that when people with IBS, they had probiotics, it improved to their stress-related GI complaints. Then we know that low food map diet also related with more anti-inflammatory properties can help to alleviate IBS symptoms. And recently, the fecal microbiota transplantation from non-IBS individuals, when we give the FMT to the IBS people with IBS, it improves their condition. And the potential mechanism how this diet and gut affecting gut microbiota and gut microbiota affecting the gut-brain axis in IBS is through the anti-inflammatory pathway. And like throughout the presentation, I've mentioned that there are quite a lot of gaps in our understanding about the gut microbiota and brain axis, because currently we don't really understand the exact neural circuit. And there are quite a lot of different techniques that we can use to study the gut microbiome and brain axis. I'm not gonna go into each if you are interested, then we can an answer session. But just a broad overview, there are currently like 7 techniques, such as the germ-free mice, and we've got antibiotics treatment. We've got the fecal microbiota transplantation, where we can use this FMT actually to simulate human gut microbiome in animal models. Then we have the prebiotics and probiotics, also psychobiotics, where we can see different treatments of diet related to the gut microbiome can actually influence the gut-brain axis. Then we have the imaging and bioinformatics that help us to directly see what happened in the brain or the nervous system when we introduce, for instance, prebiotics or probiotics to the animal models. And of course, the bioinformatics, it's very obvious. It helped us to understand the profile of the gut microbiome from individuals or samples. Then just a quick summary that currently there are quite a lot of things that wait for us in the future in terms of understanding more about the microbiota and gut-brain axis. With the current gut microbiome projects running worldwide, we will expect to have a database and better understanding about the composition of the gut microbiome in human or animals, of course. And then we can also develop new technologies by combining or improving the current one to help us to understand the neural circuits that connect the microbiome and the gut and the brain. Then, of course, the ultimate goal of studying all of this is to develop a therapeutic approach targeting the gut microbiota to help us improve our health and well-being. And just a quick summary about this talk today, that microbiota gut-brain axis is a bidirectional relationship. It involves 3 different pathways: the immune, endocrine, and neural pathways. And of course, there are many signaling molecules that involve in each pathways. Then there are crosstalks between this communication. And gut microbiota is very dynamic. It can be influenced with many factors, external, internal. And when the homeostasis of gut microbiome is changes, and when dysbiosis happens, it can impact on our brain and health condition. And this lead us to a very nice point where this gut microbiota and brain axis could be a future potential target for therapy for many diseases involving this axis. So yeah, I think that's all my presentation. I would like to thank everyone and of course Professor Amanda Page and Associate Professor Richard Young, who helped me to develop the presentation, and of course Corpus Curiosum and all the organizers and University of Adelaide, Sam Rian, the Australia Award Scholarship.